Welcome to Shaping Science, a collection of videos from the voices that are shaping science today. I'm Nikki Spahich from The Scientist, and today we're highlighting the groundbreaking technologies that won our 2025 Top Innovations contest. After perusing numerous nominations for cutting-edge products, an independent panel of judges selected the most exciting technologies for scientists from the laboratory to the clinic. Here are the winners in the Biotechnology and Healthcare and Diagnostics categories.
Cas12l is a type 5 CRISPR Cas nuclease, but it's very distinct family. From the size perspective, Cas12l is much smaller than the usual Cas9 that a lot of researchers and companies use. It's 850 amino acids, which, almost twice smaller than what's out there usually in the market. It's a nuclease that, through its PAM recognition and other characteristics, is really efficient in the editing efficiency in different cell types and can be delivered by different formats and delivery methods. We as a company come from Lithuania, and it's a country really having a great gene editing molecular biology ecosystem. From the nomenclature, we got the letter L for our Cas12, but L also stands for Lithuania. So we are proud of having a very unique tool from a really fast-growing country, which plays an important role in this CRISPR gene editing space.
Fundamentally, the challenge with science is that we don't understand the context of the words that define biology. The words being genes. If we were to look at a profile of mutations and we all had a single gene mutation in, let's say, RAS, which is a known oncogene, I would be hard-pressed to diagnose you with anything because I wouldn't know contextually what that caused. Biology is challenging because of the fact that there are so many words and contextually we can't assemble what they mean with one assay. What a VD24 is a step towards solving that problem. So with one assay, you can start to read and write the stories of biology. The technology creates an opportunity for the investigator to look at multiple aspects of biology, which include the morphology of a cell or a tissue, proteins and phosphoproteins, and it also allows you to look at the RNA in the cell and potentially even the DNA. And so with those combined pieces, a VD24 is an instrument that allows you to look at millions of cells and potentially read their stories in a single run in a 24-hour turnaround time.
Beacon Discovery is a platform for biologists. We're learning that most diseases are associated with cellular dysfunction. What the Beacon platform allows our customers to do is to directly measure cell function. Furthermore, you can link that cell function directly to the sequence. And so the biologist can understand the key molecular mechanisms that are underpinning disorders to create therapeutic breakthroughs. Today, when you think about biology, it's just overflowing with data, but many of our biologists are still struggling with insight. The question that the biologists have often is, what do the cells actually do? Our methodology is that we try to measure things directly rather than to infer things. Instead of saying, this has a CAR-T construct on the surface, it must be capable of killing a tumor cell. We'll just measure directly, and we will put a T cell and a tumor cell in a nanopen. We will videotape as the interaction between these two cells occurs, and we'll measure cytokine secretions and surface proteins, changes in those cells during that interaction. And one of our core missions is really to empower biological research. We think about the technologies that we're bringing to market. How can we help accelerate the rate at which these translational therapeutics get to patients as we continue to increase the power of what we can measure by, again, measuring these functions directly with single cell resolution. We believe that we're helping our customers get down that path faster than they could have without our technology.
UG100 is the sequencing platform sold and supported by Ultima. In February of this year, we announced a increased capability that we term Solaris. For the same amount of time for a sequencing run, we increased the output of data, the output of sequencing reads, by over 50% and reduced the price per data point by 20%. Solaris technology provides the opportunity to do a sequencing run that produces over 10 billion sequencing reads. Each of those reads is 300 bases in length. So 10 billion reads of 300 bases each on a single run, on a single silicon wafer. That price of doing a sequencing run is $2,400. On that wafer, you could sequence 30 human genomes to 30x coverage. And if you divide $2,400 by 30 samples, you would get $80 a sample. Customers around the world can access Solaris technology through Ultima's Certified Service Provider, or CSP, program. Since the time of the announcement of Solaris in February of this year, we've added several more commercial and academic service providers to our network. So we're thrilled to continue to see this expansion of very capable labs offering services using Ultima.
We're really trying to use our blood tests to estimate the presence or absence of amyloid plaques in the brain, which is a hallmark of Alzheimer's disease, right? So these amyloid proteins cause these sticky clumps in their brain and cause all kinds of problems. We discovered two biomarkers that worked really well and complemented each other, and is p-tau217 and one of the amyloid proteins, amyloid beta 42. So in combination of these two biomarkers, we can detect the presence or absence of amyloid pathology just as well as PET or CSF. What's really exciting that has come about is the FDA approval of amyloid-targeted therapies. So what this test can really provide, now that you have accessibility with a blood test, is getting these patients identified earlier in the process of this Alzheimer's disease trajectory. So if we can identify the right patients at the right time and get them on these treatments, we can definitely make a difference in their disease path. So it's really exciting to have, in combination with our pharma partners, identifying these patients earlier in their disease process.
Haystack itself is really a workflow for measuring ctDNA in a patient with exquisite sensitivity and specificity. We primarily focus on plasma derived from whole blood, and that seems to be the most abundant reservoir for cell-free DNA and circulating tumor DNA molecules from a patient's tumor. Circulating tumor DNA is a very rare phenomenon in patients with cancer, and so one of the challenges facing ctDNA technologies is how do we identify ctDNA molecules that are at very low abundance in circulation with high accuracy. One of the unique properties of Haystack MRD is its inherent error correction that's built into the workflow, which accounts for any noise that's introduced through amplification process when we're preparing the DNA or the sequencing instrument. We account for each moment in the workflow, any errors that could be introduced, done so through a very exquisite mapping and development of a workflow designed specifically for ctDNA, so that we can get very accurate measurements in patients that have very low levels of ctDNA after curative intent surgeries or during treatment when we're monitoring response.
Congratulations to all of our winners. To learn more about each innovation, click on the link below. And stay tuned for our 2026 competition, coming soon. Thank you for watching. Together, we are shaping science.